This radiograph of a bilateral hip replacement illustrates the sheer scale of the metallic surface area introduced during orthopedic surgery. These structures solve a mechanical problem, but they create a deep tissue vulnerability. In this micrograph, we see Staphylococcus bacteria clustered together. When microorganisms colonize the surface of an implant, they don't remain as isolated cells. They organize into structured communities embedded within a protective extracellular matrix. This matrix creates a physical barrier. Once this biofilm matures, the bacteria inside become highly tolerant to systemic antibiotics. This structured defense effectively shields them from the host's own immune system. When standard medications fail to penetrate this layer, the medical response is invasive. Surgeons must often perform debridement, or in many cases, remove the implant entirely to clear the infection. As long as the metal remains an inert surface, the bacteria have a permanent safe harbor. Chemical treatments struggle to breach what is essentially a mechanical defense. The MagClean hypothesis changes how we interact with the implant. By applying a time-varying magnetic field from the outside, the system creates an induced electrical microcurrent directly within the metallic implant itself. The primary innovation is achieving this electrical exposure at the metal-biofilm interface without using any implanted electrodes or percutaneous wires to deliver the charge. This microcurrent is designed to disrupt the biofilm through local pH changes, the generation of reactive oxygen species, and the physical breakdown of the extracellular matrix. While the biophysical theory is compelling, it requires a rigorous, gated translational architecture to prove its safety to regulatory bodies. To understand how this moves from the lab to the clinic, we need to deconstruct the 36-month MagClean master proposal, the exact sequence required to advance this device toward human trials. The program is governed by a translational pathway tracker. This system ensures that no phase of research begins until the safety requirements of the preceding gate are strictly met. We begin with physics modeling and bench verification. The first technical hurdle is correcting the misconception that these fields are intended to heat metal. This is not diarrhythmy. The goal is not to use temperature to kill bacteria. Gate 1 uses multi-physics modeling to prove the device achieves an electrical effect without the dangerous heat blooming seen in traditional high-frequency systems. These models verify that we can induce a target range of 200 to 2,000 microamperes of current across representative implant materials. At the same time, the simulation confirms a strict thermal boundary, maintaining a tissue temperature rise of less than 1 degree Celsius. Gate 2 moves the math into the physical world. Researchers manufactured a traceable technical batch of 16 units to begin bench verification. These units were used for dose-response studies on mature Staphylococcus aureus biofilms grown on stainless steel and titanium, providing a baseline for the device's actual biological activity. Before any living subject is exposed to the field, the engineers must prove the safe operating envelope observed in the lab maps perfectly to the theoretical math. Laboratory coupons cannot replicate the complexity of a living host, specifically immune responses and bone integration. Gate 3 addresses this through a confirmatory non-clinical study. Using a Wistar rat osteomyelitis model, researchers contaminated a femoral wire to observe the device's performance in living tissue. To separate the device's effect from standard treatments, the study used a randomized, blinded factorial design. This compared a sham device, MagClean alone, antibiotics alone, and the combination of the two. The primary metric for passing this gate is the reduction of colony-forming units recovered from the implant at the end of the trial. Success also depends on secondary safety endpoints, including histopathology and bone inflammation checks, to confirm the device causes no adverse local reactions. The go-no-go criterion is absolute. The trial cannot advance without demonstrating a reproducible anti-biofilm signal alongside zero evidence of toxicity. Once the rodent data is locked, the strategy moves into two parallel tracks: veterinary clinical investigations and staged human feasibility studies. Track A is an exploratory veterinary investigation at Fratelli Veterinary Hospital. This stage involves 36 cats and dogs with existing metallic implants. The focus here is strictly on safety, tolerability, and feasibility in a clinical setting. At this stage, researchers avoid making broad claims of a cure, focusing instead on how well the animal subjects tolerate the daily sessions. Track B is the human progression. Governed by ISO standards, this track is divided into 3 stepped stages, each with its own safety barrier. Stage 8A begins with healthy volunteers who do not have implants. This allows researchers to verify skin contact safety and thermal limits without the variable of an internal metal device. Stage 8B expands to adults with stable, uninfected implants. This verifies the system's logging, alarms, and stability when interacting with in situ metal. Gate 5 is the first inpatient study. This is a sentinel cohort, an initial group of just 5 adults suffering from chronic implant-associated infections. During this phase, standard medical care is never withheld. The system is evaluated as a non-invasive adjunct to the patient's existing antimicrobial therapy. Human clinical design is not a single leap. It is a stepwise retirement of risk, designed to protect patients at every increment of the process. This 36-month translational pipeline completes its arc at Gate 6, where the collected data forms the foundation for a final medical device. We have traced the evidence from electromagnetic simulations and benchtop biofilms through factorial rodent models and veterinary patients, arriving finally at sentinel human cohorts. As the master proposal notes, program success does not require a positive result for every single hypothesis. Real R&D success is the generation of reliable, traceable data. It provides the objective basis to decide whether to advance, iterate, or stop. If this 6-gate pathway holds true, it changes the role of the implant. It takes the very metal that allows the bacteria to hide and turns it into a therapeutic electrode to destroy them.